Welcome to this lecture on evolutionary biology. Today, we are going to explore one of the most fascinating and powerful concepts in modern science, known as the molecular clock. Have you ever wondered how scientists know exactly when humans and chimpanzees split from a common ancestor, even without a perfect fossil record? The answer lies hidden right inside our cells, written in the language of DNA. By the end of this video, you will understand how to read this biological timepiece. To understand how this biological timepiece works, let us start with a simple analogy. Imagine that you and a friend buy identical, brand new notebooks on the exact same day. Every single week, without fail, you each randomly scribble on one page of your own notebook. If we meet you years later and count that there are exactly 104 scribbled pages in your notebook, we can easily calculate that you have owned it for 104 weeks. In evolutionary biology, DNA acts exactly like this notebook, and random genetic mutations are the scribbles. Because these mutations happen at a relatively steady and predictable rate over millions of years, we can simply count them to figure out how much time has passed. Now that we have our notebook analogy, let us look at the actual biological mechanism behind the molecular clock. At its core, this technique relies on the observation that certain biomolecules, like DNA sequences, experience neutral mutations at a constant rate over geological time. A neutral mutation is simply a change in the genetic code that neither helps nor harms the organism. Because it is harmless, natural selection does not weed it out. These changes pile up steadily, like a ticking metronome. By comparing the DNA of two living species and counting the number of differences between them, scientists can mathematically trace back the branches of the evolutionary tree to find out exactly when those two species split apart. Understanding this genetic mechanism naturally leads us to ask why we even need a molecular clock when we can just dig up fossils. The truth is that the physical fossil record is incredibly incomplete. Most organisms decay entirely without leaving any trace behind in the rocks. The molecular clock fills in these massive gaps. It allows us to date evolutionary events for soft-bodied creatures, microscopic bacteria, or species living in environments where fossils simply do not form. In the real world, epidemiologists even use this tool to track the origins of deadly viruses like HIV or SARS-CoV-2, helping us understand when a disease jumped from animals to humans. Since we now know why this tool is so essential, let us look at the fundamental equation that makes it work. To use the molecular clock in practice, we need a mathematical relationship connecting genetic differences to actual time. The formula is T equals D divided by 2R. Let us break down what each symbol means. The letter T represents the time that has passed since the two species diverged, usually measured in millions of years. The letter D stands for the total number of genetic differences observed between the two species today. The letter R represents the mutation rate, which is the number of genetic changes per million years in a single lineage. We divide by the number 2 because both species have been evolving and mutating separately since they split. If the mutation rate increases, the calculated time for a given number of differences will decrease. Before we move on to more complex applications, let us see this formula in action with a concrete example. Imagine we are comparing a specific gene between a human and a chimpanzee. Scientists find a total of 30 genetic differences between the two species. Previous studies established that the mutation rate for this gene is 2.5 mutations per million years per lineage. We want to find the divergence time. We substitute 30 for D and 2.5 for R. The denominator becomes 2 times 2.5, which equals 5. This means the two lineages combined accumulate 5 mutations every million years. Dividing 30 by 5 gives us a final answer of 6. Physically, this means the evolutionary lineages leading to modern humans and chimpanzees split from a single common ancestor exactly 6 million years ago. Now that we have successfully used the standard formula, we must address a common problem. What if we do not know the mutation rate? To solve this, we need to derive a calibration formula. We start with our standard equation, where time equals differences divided by 2 times the rate. Our goal is to isolate the mutation rate. First, we multiply both sides of the equation by 2 times the rate. This brings the rate out of the denominator and over to the left side, giving us 2 times the rate times time equals the differences. Finally, we divide both sides by 2 times the time. This leaves the mutation rate completely isolated on the left. We have now proven that the mutation rate equals the total differences divided by 2 times the divergence time. This simple algebraic rearrangement is incredibly powerful. This brings us to the formal calibration equation we just derived. The formula is R equals D divided by 2T. Conceptually, this allows us to use a known fossil to find how fast the genetic clock is ticking for a specific group of organisms. The symbol R is the mutation rate we want to find. The symbol D is the total number of genetic differences we count between the two species today. The symbol T is the known divergence time provided by the fossil record. If the known divergence time T increases for a fixed number of differences, the calculated mutation rate will decrease, meaning the clock ticks more slowly. With our newly calibrated formula clearly defined, let us apply it to a real-world scenario using fossil evidence. Suppose a well-preserved fossil tells us that species A and species B diverged exactly 10 million years ago. When we compare their DNA today, we find 40 genetic differences. We need to calculate the unknown mutation rate. We take our calibration formula and plug in the known values. The total differences, D, is 40. The time, T, is 10 million years. The denominator becomes 2 times 10, which is 20. Dividing 40 by 20 gives us exactly 2. This means the genetic clock for this specific group of organisms is ticking at a constant rate of 2 mutations per million years per lineage. Finding this rate is the critical first step to unlocking the rest of the evolutionary tree. Now that our molecular clock is perfectly calibrated, let us use it to discover something entirely new. Imagine we discover a third organism, species C. When we compare its DNA to species A, we find 60 genetic differences. Because we already calibrated the clock, we know the constant mutation rate is 2. We simply plug our new difference of 60 and our calibrated rate of 2 back into the original time equation. The denominator is 2 times 2, which equals 4. Dividing 60 by 4 gives us 15. This physically means that species A and species C shared a common ancestor 15 million years ago. By using a single fossil to calibrate the clock, we successfully uncovered the hidden evolutionary timeline of a completely different species. To visually reinforce what we have just calculated, let us look at a phylogenetic tree diagram. On the vertical axis on the left, we have time, measured in millions of years, moving forward as we go up. At the very bottom, the single line represents the common ancestor. The point where the line splits into a Y shape is the exact moment of divergence we have been calculating. The left branch leads to species A, and the right branch leads to species B. Notice the small perpendicular tick marks along both branches. Each of these marks represents a single neutral genetic mutation accumulating at a steady rate over time. This diagram beautifully illustrates how both lineages evolve and mutate independently after they split. Having seen the mathematics and the visual representation, we can truly appreciate the predictive power of this biological tool. The molecular clock is an elegant and indispensable technique that literally turns DNA into a historical timepiece. Whether we are tracing the ancient origins of humanity across millions of years or tracking the rapid spread of a modern virus over a few months, the underlying mathematics remain exactly the same. It ensures that we can map out the massive, complex tree of life and understand our biological history, even when the fossil record is completely silent. It proves that biology and mathematics work together perfectly. We have covered a lot of ground today, so let us summarize the key takeaways. First, remember that DNA accumulates neutral mutations at a constant, predictable rate, acting just like a ticking clock. Second, we can calculate the exact divergence time between two species using the total genetic differences and the mutation rate. Finally, when the mutation rate is unknown, we can use known fossil evidence to calibrate the clock. You now understand the fundamental principles of the molecular clock, a tool that is absolutely essential for modern evolutionary biology.